Photosynthesis, nature's recipe. Have you ever wondered what type of food plants eat or how they make it? Well, it turns out that they're pretty good chefs and they can make their own food via a process known as photosynthesis. Photosynthesis can be broken down into two words: photo, which means light, and synthesis, which means to make things from components. In photosynthesis, carbon dioxide and water are used to make a molecule of glucose plus a byproduct of oxygen. This reaction requires sunlight and a pigment molecule known as chlorophyll. Here is the chemical equation of photosynthesis. 6 molecules of carbon dioxide plus 6 molecules of water is converted into 1 molecule of glucose plus 6 molecules of oxygen. If you look back to the structure of a plant cell, you will remember that there's a cellular structure known as chloroplast. This acts like a kitchen for the plant cells, as it's the location for the first stage of photosynthesis. Here is a closer look at the structure of chloroplast. It has an outer membrane, an inner membrane, thylakoids, stroma, which is luminal space, and a stack of thylakoids is known as granum. Chlorophyll is located in a protein complex that spans the thylakoid membrane. The sun shines different wavelengths of light onto the chlorophyll in the chloroplast of the cell. The pigment molecule chlorophyll is able to absorb the different wavelengths of light except for the green wavelength in which it reflects. And this is what causes leaves to appear a green colour. Water absorbed by plants is split into protons, electrons and oxygen. The protons is stored in a molecule known as NADPH, and the electrons are stored in a form of ATP. A tube-like tissue known as xylem transports the absorbed water from the roots in an upward direction, in a similar way as a straw sucks up water from a cup. The transported water is delivered to the leaves. The leaves of a plant have pores known as stoma. The stoma can either be open or closed, depending on the swelling of the guard cells. Gas exchange in plants occurs in the stoma. When the pore is open, carbon dioxide is able to enter the cell, and oxygen is able to leave the cell. We have seen how oxygen is released from plants during the light-dependent stage. Now let us look at how plants use the carbon dioxide that they uptake from the atmosphere. Carbon fixation is the process where inorganic carbon dioxide is converted into organic carbohydrate, glucose. It's a part of a cycle known as the Calvin cycle. And before you say it, this has nothing to do with Calvin Harris. The Calvin cycle occurs in the stroma of the chloroplast and requires a number of ingredients including carbon dioxide, a 5-carbon molecule ribulose bisphosphate, an enzyme rubisco, and the NADPH and ATP from the light-dependent reaction. The first stage of the Calvin cycle is carbon fixation. This is where 1 molecule of carbon dioxide is fixed onto the 5-carbon molecule using the enzyme rubisco. This produces an unstable 6-carbon intermediate molecule. The unstable 6-carbon molecule is split into 2 identical 3-carbon molecules. The 2 3-carbon molecules, known as 3-PGA, are reduced by using a hydrogen from NADPH and also phosphorylated using a phosphate from ATP. For every 3 ribulose bisphosphate, 6 G3Ps are made. 1 out of 6 of the G3Ps are used to make glucose, while 5 out of 6 of the G3Ps are recycled back into the carbon cycle. This means that 2 carbon cycles are required to make 1 molecule of glucose. Here is an overview diagram of the carbon cycle. Algae is the name given to a group of mysterious photosynthetic organisms. We're not quite sure whether these organisms are plants or not. You can see algae forming on the top layers of ponds or in swampy lakes, and it's quite gooey. Or you can see these on the bottom of the seabed covering coral. Although it doesn't look quite attractive, algae can be pretty useful. It can be used as fuel to run our cars and homes, and is also edible. Blue tangs, like Dory, eat the algae on coral to ensure that the coral on the bottom of the seabed doesn't suffocate. The rate of photosynthesis can be measured by either the uptake of carbon dioxide or the release of oxygen. Rate equals the amount of gas either taken up or released over a certain period of time. The rate of photosynthesis is dependent on many factors, including the presence of light, light intensity, the wavelength of light, concentration of carbon dioxide available, and temperature. All of these factors can be investigated experimentally.